that of the person, their research discipline, their research sector, and whatever it may be. So first I am amazed what to talk about the aspects of research. Then I decided and narrowed to share what type of research, in what aspects we are carrying out in the research in our lab. So that I decided. From that point of view, I would like to share the aspect of our research, what we are doing in our lab, in our department. We are mainly focused on, mainly working on microbiological research with respect to infectious diseases. In recent days, we are working on nanotechnology approaches for developing some sort of technologies for the combating of infectious diseases, especially with respect to the combating microorganisms. That is what I'm going to share with this colloquium. The title of my talk is nothing but Green Nanoscience, a natural product-based nanoformulations for combating infectious disease. So first, let me introduce about nanotechnology. So we all know about nanotechnology. It is an emerging discipline which have a potential for the generation of new and novel innovating materials. Today, nanotechnology has the ability to open new avenues in multiple disciplines, such as in all the disciplines, including environment, solar energy, conversion, catalysis, medicine, and whatever it may be, and wastewater treatment. The reason behind these versatile applications of nanobiotechnology, especially nanomaterials, is nothing but their unique properties. That nanosize, that size matters. These unique properties of nanoparticles or nanomaterials are receiving increasing attention in biomedical applications, such as biological sensing, antimicrobial activity, medical diagnosis, cancer therapy, drug delivery, and artificial intelligence. There are a lot of applications for nanomaterials. I hope so. In all the institutions, at least there will be a person who works on nanomaterials. So this is the hot topic in recent decades. And that's why I've decided to share the aspects what we are doing, what aspects of research we are doing, performing in nanotechnological research. So that will be suitable for these colloquium meetings. Okay. But there are a lot of nanomaterials available. There are different types of nanomaterials, right from metal nanoparticles, polymer nanomaterials, synthetic nanomaterials that includes some sort of synthetic materials. But our research, what we are doing in this sense, we are focusing on green nanoscience. That means we are using the herbal medicine, especially herbal-based nanomaterials. We are developing herbal-based nanomaterials for the combating infectious disease in multiple aspects. So the reason why we have chosen that herbal medicine as a theme for our research, the thing is, in recent decades, especially now we are in a COVID pandemic situation. So this is a good example for us. Whatever situation is there, our health is very, very important. Okay. So if we have some sort of diseases, we are naturally depending upon some sort of natural materials, especially immediately we rely on plant materials for the cure. Okay. It is not a present situation. So, human dependence on plants as a source for medicine, it dates back from prehistoric times. Even now, more than 3/4 of the world population mainly rely on plant products for their therapeutic purposes and healthcare aspects. Even WHO, it has been estimated that 80% of populations in developing countries depend upon, they highly rely on traditional herbal medicines for their healthcare needs. It has been recently cited. And more than, whatever may be allopathic cases, more than 620 drugs predicted from aliments are derived from plants and several other natural plant-based compounds. Even that, for example, a good example for the unique antibiotic, streptomycin. It is not a plant-based, but it is a natural-based one. Okay. In recent times, nanomaterials is not new for us. It is a very old method. So green chemistry plays an important role in the field of nanoscience and nanotechnology, especially in the fabrication of nanoparticles. For multiple aspects, we are going to narrow it on therapeutic applications. And one positive effect of these herbal materials are they are eco-friendly, they are non-allergic. Non-allergic, they are less allergic and less toxic, but possess considerable issues such as salt durability, and low stability under ambient conditions. Nanoization of the phytochemicals results in enhanced permeability and retention effect. It also helps in the tissue distribution effect, macrophages, sustained release, and further physiochemical stability is highly questioned. So in this aspect, normal phytochemicals have some sort of side effects like this. So we decided to apply nanotechnology on these aspects with respect to the herbal medicines. Instead of giving a theoretical lecture, just I would like to present our work, what we have performed in our lab. I'm going to present 3 works what we performed in our work. In that aspect, I can explain what aspects of research we are doing in this topic. Okay. First one is the title of our work is Fabrication and Characterization of Ficus racemosa latex biopolymer and Acalifa plant extract loaded nanofibrous matrix for biomedical applications. So here, what we do is introduction to this work is nothing but wound infection. We have developed a nanomaterial for wound healing with herbal medicine based approaches. So wound infection remains a significant problem with considerable morbidity and mortality worldwide. So it is been estimated that 1 to 2% of the world population in developing countries are affected by the chronic wounds. Okay, so in order to treat the chronic wounds, we are applying antibiotics. But antibiotics have conferred selective advantages for naturally resistant bacteria, which simultaneously leads to the drug ineffectiveness. Okay, that's what happened in the recent decades. Most of the drugs, the microbes have developed resistance. So now we are in the search for novel classes of antibiotics. Now most of the biomedical research is being diverted toward the development of novel classes of antibiotics. So, and next one is if we have a wound infection. In olden days, what we do in this, in traditional cotton we apply traditional cotton resins. But these traditional cotton resins fails to ensure complete protection from infection, and also simultaneously it delays the wound healing process. It delays the wound healing process, and there is a emergence of antibiotic resistant pathogens, and these antibiotic resistant pathogens have received an interest in scientific community to develop alternative methods to reduce these bioburdens in healthcare facilities, to search a new cost-effective drugs from natural origin. Okay, this is what we are doing. So herbal medicine is still an important component of healthcare in India. Recently in 2014, the central government have initiated a ministry called as Ayush. Ayush it is an acronym. Ayurveda, Yoga, Siddha, Homeopathy. So that is called as Ayush. They have initiated Ayush to popularize and to follow and to implement our traditional herbal medicine for the treatment of different diseases. Besides the classical system of Indian medicine, there are a lot of medicines, folk, tribal medicines, a lot of plant products have been traditionally used for the treatment of wounds and burns. Okay. Several natural products of plant origin are described in traditional text of Indian system of medicine for their healing properties under the term Ranaropaka. So this is an Ayurvedic term. So in our literature servers have also cited the application of plant-based products in our traditional systems of medicines. When we talk about wound healing aspects, whatever it may be, we can't eliminate the role of polymers. So polymers are very, very essential for the heat treatment of wound healings. Natural polymers are widely used in the field of regenerative medicine. They have been used as wound dressing and healing materials due to their high biocompatibility and biodegradability. And these natural polymers also act as a similar to extracellular matrix of the human skin. Recent studies have suggested that natural polymers exhibited strong antibacterial activity. Certain natural polymers, not all the natural polymers, they exhibited strong antibacterial activity and promotes wound healing by reducing the cytokine release. They reduces inflammation, decreasing lymphocyte and mast cell infiltration. Natural molecules show, and further, these natural polymers show a relatively low mechanical strength compared to the synthetic polymers. But these sort of issues can be improved by cross-linking or blending these natural polymers with that of the synthetic polymers to improve the mechanical property and other. This is what current wound healing research is being performed. Okay. Next one. So the thing is, first we decided to have a nano-based approach for the treatment of wound healing process. For that, we decided to select a natural-based polymer. For the source of a natural-based polymer, we have a diverse literature survey, and we identified a particular plant that is called as tree ficus racemosa. In Tamil, அது அத்தின்னு சொல்லுவோம், அத்திமரம். So ficus racemosa. These ficus racemosa have been the medicinal application of this ficus racemosa has been cited in many literatures. Okay, it is a popular medicinal plant of India, which has been long used for the treatment of various diseases and disorders. So all the parts of this leaf, this plant is like leaves, roots, bark, they are indicated as medically important in all the most of the traditional system medicine, traditional Indian medicine, Ayurveda, Siddha, whatever it may be. Okay. But in this study, what we did is that we focused on we isolated the latex. பால்னு சொல்லுவாங்க, அத்தி மரத்தை லேசா கொத்துனீங்கன்னா பால். So that latex. So it has been கலித்தொகை. தமிழ்ல கலித்தொகைன்னு ஒரு நூல் இருக்கு. அதுல they have been cited. போர் இதுல வந்து என்ன சைட் பண்ணிருக்காங்க அப்படின்னு பாத்தீங்கன்னா, அத்தி மரத்தினாலான உலோகங்களால் ஏற்படும் புண்களுக்கு அத்தி மரத்தின் பாலை தடவினால் சரியா போகுதுன்னு சொல்லி கலித்தொகையில ஒரு சைட்டேஷன்ஸ். ஓகே, உலோகத்தினால் ஏற்படும் ரணங்களுக்கு அப்படின்னு ஒரு கான்செப்ட். ஸோ வி டிசைடட் அதுல கலித்தொகையில இருக்கு. ஸோ கலித்தொகையில உலோகத்துல அந்த காலத்துல மெட்டல். ஸோ மெட்டல் ஏஜ் நம்ம ஹியூமன் எவல்யூஷன் அப்படின்னு பாத்தீங்கன்னா மெட்டல் ஏஜ் அப்படின்றது ரேர். ஸோ அப்போ அந்த காலத்துல they have cited about metals. ஸோ அப்போ மெட்டல்ஸ் அண்ட் they have correlated the metals and woods. அப்போ இங்க எப்படி இருக்கும் அப்படின்னா நத்திங் பட் இன் வார், போர்க்களத்துல உலோகம் அங்கதான் பயன்படுத்துறதுக்கான பாசிபிலிட்டிஸ். ஸோ அப்போ அந்த போர்ல if they have some sort of scars, they would have been damaged due to some metals, கத்தினால இதா இருக்கும். ஸோ அதுக்கு வந்து at that time they applied these latex of ficus racemosa. அத்தி மரத்தோட லாட்டெக்ஸ் எடுத்து அப்ளை பண்ணி they have cured the wounds. So that is the one for our research. So what we decided, so we can start with this point. We decided to start with this point. So we isolated or we retrieved the latex from ficus tree. Okay, ficus நிறைய species இருக்கு. அதுல ficus racemosa species நாங்க டெவலப் பண்ணோம், which is available in our campus. So we decided to take the latex from ficus racemosa tree, and it has been cited. We performed literature survey. அது வந்து பாத்தீங்கன்னா in 1999 these Yadav have reported that these latex have been applied for the treatment of boils, scars and lesions. Okay, so and also ficus racemosa latex is being used for the treatment of skeleton fracture in indigenous system of medicine in Sri Lanka. Not only in Indian medicine, Sri Lankan traditional medicine, these wound healing related approach for ficus medicine already. So we decided to these ficus racemosa latex as a source for the isolation of polymers. Then, so for wound அப்படி இருக்கும் அப்படின்னு பாத்தீங்கன்னா, not only wound healing is an ultimate aim, but if there is an open wound, the healing is not at all a matter. Healing is also a matter, but at the same time, when there is a wound, there may be a sort of infections may occur in the wound. So அப்போ wound healing அப்படின்றது infectionsனால wound healing rate வந்து பாதிக்கப்படும். So in formulating a product, that natural polymers will they facilitate the healing of wounds. In addition to that polymers, we should add some sort of natural products, plant extracts. We should add some sort of natural extracts which prevent the wound from infection or from the bacterial infections. For that, we decided to choose Acalypha indica, Kupamine. எல்லாருக்கும் தெரியும். சின்ன folk medicine, traditional medicine, சின்ன வயசுல நமக்கு ஏதோ அடிபட்டுச்சுன்னா they will use usually கீழே விழுந்து சராய்ச்சி வச்சுன்னா என்ன பண்ணுவாங்க அப்படின்னா குப்பமேனி எடுத்து தெரியுமா, குப்பமேனி applications நிறைய literature சைட் பண்ணிருக்கு. So Acalypha indica, Euphorbiaceae family, there are more than 124 species. It is one of the important herb cited in herbal medicine, both in Siddha and Ayurveda, for the treatment of more than 70 human diseases. Okay, the health benefits of Acalypha indica, especially with skin care, has been highly reported. And it's also possessed to have anti-inflammatory activity, antioxidative activity, antimicrobial, venom neutralization, and so on. Okay, the all part of leaves, root, stalk, and flower have been used for the treatment of various skin-based disorders and multiple medicinal properties. So, in this study, we decided to take the polymer from ficus racemosa tree for nano-based approaches, and we have chosen Acalypha indica plant extract for antimicrobial activity. Now we are applying these two aspects in nanotechnology. What we are doing is there is a process called as electrospinning. So electrospinning is a very simple and versatile method that applies high voltage electrical field to fabricate nanofibers with large surface area to volume ratio. These nanofibers will have some sort of interconnected cores. Here you can see the nanofibers. This is a scanning electron microscopy of nanofiber. So electron spun nanofibers resemble, and another very important thing is these electron spun nanofibers resembles a natural extracellular matrix and promotes proliferation of cells. Okay, recent studies have focused on incorporating this natural extract with polymer-based electrospun nanofibers for biomedical applications. So what we decided is, for performing this electrospinning and fabricating a nanofiber, we need to have a polymer and a plant extract. So for that, we isolated polymer from Ficus racemosa latex, and for antimicrobial activity, we taken the Acalypha indica leaf extract. We blended these two and we applied it for electrospinning, and we fabricated a polymer, fabricated a nanofiber. These are our objectives. So I've already mentioned it, and these are the characterizations. Let me explain all the reasons. Okay, these are our objectives, and these are our methodology. We have performed, we have isolated the polymer, we have characterized the polymer, we have fabricated the nanofiber, and we have tested the biocompatibility analysis that includes hemolysis test, blood clotting index, cytoplasmic activity, wound fluid absorption studies, antimicrobial and in vivo wound healing activity. So these are our results. This is the crude latex that has been isolated from the Ficus racemosa tree, and from that we have isolated this polymer. So this is a polymer. We have purified this polymer, and we have confirmed the polymer that is not a starch. In plants, most of the plants predominant polymer is nothing but starch. So carbohydrate polymer, we have to differentiate whether we have isolated a carbohydrate polymer or a starch, whether it is starch or not. For that, we have performed the basic test, smallest test, and added test. From that assay, we have confirmed the isolated polymer is not a starch. Okay, not a starch. Then we have went for solubility test. The isolated polymer from the natural polymer from the latex of Ficus racemosa is highly soluble in water, and they are insoluble in other non-polar solvents such as ethanol and acetone. Then we have taken the UV visible spectroscopy for confirming the stability and backbone. So we have received the peak at we notified a peak at 270 nanometer that correspond to pi-pi transition of polymeric backbone. A peak at 330 to 340 indicates a n-pi transition of the polymeric matrix. So these evidences confirm these two results, and have confirmed that the isolated material is a polymer other than starch, and we have identified with the help of UV spectroscopy of the polymer, and further we identify what are the functional group present in the polymer. Okay. Functional group. Then we went for NMR spectroscopy, and all these, and we have find in the NMR spectroscopy the presence of glycosidic rings, and it again indicates the existence of it is a heteropolysaccharide with alpha configuration. So all these physical tests confirm the isolated polymer, natural polymer is a polymer other than a starch. Okay, and in order to going for application, the polymer stability is a very, very important criteria. So we decided to elucidate the thermal stability of a polymer. So we have performed thermogravimetric analysis. The inference of the thermogravimetric analysis confirms the polymer structure is highly stable up to 300 degrees Celsius. For biomedical applications, we will be applying only for 30 to 40. That is a maximum limit. But these polymers have stability up to 300 to 400 degrees Celsius. So it confirms the polymer is highly stable. Therefore, we can go for further analysis. And X-ray diffraction also confirms the crystalline nature. So we have also confirmed with the X-ray diffraction association. And this is what the polymer, that the nanofiber we fabricated with the polymer, natural polymer that has been isolated from Ficus racemosa latex, and we have incorporated the polymer with that of the Acalypha indica plant extract. இதுதான் வந்து fabricate பண்ண polymer உடைய eye view, and this is nothing but scanning ultraviolet. This polymer will have some sort of pain and it resembles our skin. If we focus, if we zoom our skin, it will resemble like that. இந்த மாதிரி ஒரு மேட்ரிக் சிஸ்டம் இருக்கும். ஸ்கின்ல ஏர் transfusion மற்ற வாட்டர் டிஃப்யூஷனுக்கு சின்ன சின்ன போர்ஸ் இருக்கும். ஸோ திஸ் நானோ ஃபைபர் will resemble our skin, but the only one important feature is it is a nanofiber which has nanopores. அப்ப என்ன பண்ண முடியும்னா microbes are bacteria are microbes, they will have only microstructures. So since the pores have nanostructures, external infection from the environment may be prevented with the help while applying these nanofibers for the treatment process. Okay, so this is our fabricated electrospun nanofiber based on natural products, and we assess the biocompatibility assay. So it is highly biocompatible. Okay, it is highly biocompatible, and anti-thrombogenic activity also we found in this polymer. It has about 81% of enhanced anti-thrombogenic activity. And the next one is cytotoxicity. We have also performed different blue assay for analyzing whether these nanofiber or Acalypha indica plant extract have some sort of cytotoxic activity. It doesn't possess any toxic effect. It has been confirmed with the help of different blue assay, and these are the antimicrobial activity of the fabricated nanofibers. So, and the fabricated nanofibers punch panni we have tested with different microorganisms. It possesses both, has the ability to control both bacteria and fungus. So it has a potent multiple antimicrobial activity. Okay, adukapra vandu pathina an important aspect for better biomaterial, especially for wound healing. Wound fluid absorption studies. If it is a normal wound, there won't be any problem. Sometimes an wound may be an exudate wounds. Exudate wounds அப்படின்னு பார்த்தீங்கன்னா ஃபர்ஸ்ட் வந்துக்கிட்டே இருக்கும். ஸோ அந்த மாதிரி wound க்யூர் பண்ணுறது ரொம்ப கஷ்டம். அப்போ அந்த மாதிரி wound நம்ம மெடிசன் அப்ளை பண்ணாலும் இல்லை பேண்டேஜ் அப்ளை பண்ணாலுமே வந்து ரொம்ப நேரம் அங்கே இருக்காது. ஸோ wound healing rate வந்து ரொம்ப கம்மியாக இருக்கும். அப்போது ஃபார் டிசைனிங் a better wound healing agent, அந்த பாலிமருக்கு என்ன கெப்பாசிட்டி இருக்குன்னா அந்த wound fluid absorption capacity should be high. So that is one of the important criteria in designing or developing biomaterial for wound fluid. So abhi paakuvilla. So we have performed a simulated wound fluid absorption studies in our lab. நம்ம plasma இருக்கக்கூடிய elements fulla artificially we have prepared in the medium, and we soaked that fabricated biopolymer in that medium, and different hours we have observed the absorption. எந்த அளவுக்கு fluid வந்து absorption capacity. So 72 hours வரைக்கும் they can absorb a maximum of 65% of solution what we apply. So அப்போ presence எவ்வளவு தூரம் ஸ்கின்ல அந்த நம்மளோட பயோமெட்ரிக் presence இருக்கோ அவ்வளவு தூரம் healing rate ஜாஸ்தியா இருக்கும். So at the same time, in case of treating exudate wounds, it is very difficult. So in these polymer can be applied in even in exudate wounds also. So அப்போ exudate wound லையுமே வந்து apply பண்ணலாம். Then we went for in vivo animal studies. So animal studies we get a better results. So on day 5, so control one the mat apply பண்ணது. So on day 5, wound contraction for the control was 11%, where it was 40 for ficus racemosa, this fabricated fiber. அதே மாதிரி we are also checking with that of the standard ointment dermazine apply பண்ணி பார்க்கணும். Okay, so compare பண்ணி பார்க்கறப்போ these nanofibers have a better effect in the wound healing of the wound healing process. So this is one of the good results what we observed. So this is the conclusion of our study. The present study, we have developed a novel biopolymer based nanofiber mats by electrospinning method. And these electrospun nanofiber process a better biocompatibility, superior 90 microbial activity also. So these can be formulated, and these nanofibers products can be helpful in screening, helpful in wound healing process. This is the first work with respect to the natural product based wound healing process. And this is our another work. I'm not going to present in detail. So first, for an introduction, I have detailedly explained this work, but in next work, what we do in this is Next work, we have performed the fabrication and characterization of herbal nanoparticles loaded polymeric nanofibers and microfibers. So there are two processes. Electrospinning, we can create nanofibers. In electrospraying, we can develop microspheres. Here we perform both we have developed both nanofiber and microsphere for wound healing process. Here, this time we have changed the material. We have used instead of Acalifra indica, we have used Colloquium. Is nothing but Tamil name again the choose Ranakalli. Ranakalli. Ranale wound in Natham. So Tamil naturally the pair and Ranakalli. So we decided to choose that plant. So Ranakalli. Instead of directly applying as a plant extract, அதை ட்ரை பண்ணி அதை பால் மில்லிங் ப்ராசஸ் மூலமா நானோ சைஸ்க்கு அதோட ஸ்ட்ரக்சர் ரெடியூஸ் பண்றோம். ஸோ தேர்ஃபார் we have indicated as a herbal nano particles. ஸோ நானோ பவுடர்ஸை எடுத்துட்டு அதை பாலிமர்ஸ் செல்லுலோஸ் அசிட்டேட்டோட பாலிமர்ஸோட பிளெண்ட் பண்ணி எலக்ட்ரோஸ்பின்னிங் மூலமா we have created nano fiber and microsphere. So it has a better activity than the previous one. Okay. In addition, wound healing process and antimicrobial activity. இந்த இதுக்கு என்ன இருந்துச்சு அப்படின்னா we studied the anti-cancer activity of these herbal nanoparticle loaded microspheres in skin cancers alone, because the application work is skin-based. We have tested these nanomicrospheres for anti-cancer activity. We got a best results. So these herbal nanoparticle loaded microspheres, Colloquium herbal nanoparticle loaded microspheres possess better anti-cancer activity. So this is our inference of the second work. This is one of the work, and the next work is nothing but in this work what we do in the sense we have performed carbon based nanomaterials. So graphene oxide, I hope you all know about graphene oxide. So here with the help of herbal nanopowder we have developed graphene oxides. Reduced graphene oxide. We have converted graphene oxide into graphene oxide nanosheets, and these graphene oxide reduced biologically or traditionally reduced graphene oxide process quorum sensing inhibition, quorum quenching activity. So this is quorum quenching activity. Quorum quenching is nothing but a mechanism by which the pathogen develops biofilms. So biofilm is a very, very important criteria for a pathogen. So these herbal powder based synthesis of the reduced graphene oxide process has the ability of inhibiting the biofilm formation. This is a very, very important result what we get. So here we have represented the conversion of this is normal graphene, this is a reduced graphene oxide with the help of herbal nanopowder, and this is the scanning electron microscopic view, and this is the AFM analysis, atomic force microscopy of these fabricated carbon-based nanoparticle, and here we can see the nanosheets of this process present potent quorum sensing, okay, and also possess some sort of anti-cancer activity also observed in this work. So this is our second work, and our third work. Here we have taken in the first two works we have developed nanofibers and microspheres that comes under polymer-based nanomaterials. Here carbon-based nanomaterials. The third final one is nano emulsions. Okay, here we perform tea tree oil-based nano emulsions, evaluation of biofilm inhibition by in vitro and in silico approach. Here we accessed tea tree oil, and we have converted this tea tree oil to nano emulsion. The property of nano emulsion will have high reactive property, and you can mix with that of the, and the nano emulsion will be highly stable, and they can mix with water and oil. Okay, this is nano emulsion. We have performed GCMS analysis of the tea tree oil, and these are the different components present in the tea tree oil. And we have, these are the scanning electron microscopic assay of nano emulsions. You can see these are the droplets, and this is the nano emulsion, nano in size, and DLS and dynamic light scattering and confirm the size confirmation, and this is at the nano level 0.055 nanometer. Okay, so this is where this is our result. We have successfully fabricated a nano emulsion, tea tree oil based nano emulsion, which has potent biofilm inhibition. So this is a conjugate plate method. Here in control, biofilm is formed and we applied this nano emulsion at this part. After the application of nano emulsion, biofilms are green, not formed in this one. So these tea tree oil based nano emulsions has a better biofilm inhibition activity. So these can be used in the treatment of various pathogenic infectious diseases. And the last one is, apart from doing nano-based approaches, what we did in this is we do some sort of we perform some computation analysis. So we are with the help in GCMS analysis, we have identified the structures of these compounds. In this study, we have performed some in silico studies. We have taken the target for biofilm formation. We have docked with this anti-biofilm formation target. These among the most of the beta-pinene, terpenin, terpinol, calophyllin, and cubanin, and selenine, and another compound that is called as selenine is not been incorporated. Selenine, approximately 78 compounds from this tea tree oil based components showed better anti-biofilm activity. So we confirmed the anti-biofilm action with the help of computation studies. We have developed some sort of lead organisms from that particular beta-selenine structure, and we have confirmed with the doctor. And also we have performed some homo-lumo analysis, chemistry-based analysis, and now molecular dynamic studies is in progress. So this is our research regarding natural product-based therapeutic approaches for the treatment of in silico studies. Thank you.
Scholars, if you have any doubt, you can clarify.
Any clarifications?
How you have done the wound healing activity, Mr. Karthik? Do you
Wound
have
healing
animal ethics committee under?
Yes. No, we doesn't have animal ethics committee. I have performed in IIT.
Oh, okay.
With the help of my friend.
Okay.
Any other clarifications? So this is our basic work, what we are doing in our lab. Now we are focusing on some other nanotechnological approaches. Just we have entered into nano emulsions. Next, we are trying to apply it for drug delivery approaches.
Through the application of bioinformatics, isn't it? You are planning to have.
Madam?
You are planning to have through the application of bioinformatics, drug delivery and designing.
Yes. The thing is, for example, we have from these list of compounds from the tea tree oil, we have selected a better one, that is beta-selenine, this structure.
Okay.
This structure has a potent anti-biofilm activity as an individual compound. So now what we are doing in this sense, we are trying to increase the, for example, if this beta-selenine, experimentally we are studying the experimental, we are measuring the experimental parameter. So for example, if it has a 20%, these beta-selenine have 20% biofilm inhibition activity. But now what we are doing in the sense, we are modifying the structure of beta-selenine. Already it has been confirmed both experimental and bioinformatics aspects, these beta-selenine having anti-biofilm activity. Now we are modifying the structure in computation biology. So we are including all these groups in the structure, and we are assessing whether they include what addition in this beta-selenine structure.
Enhancing its activity.
That is what we are doing now, and we are initially performing molecular dynamic studies, and we are also performing some chemistry-based analysis for highest occupied molecular orbital and lowest occupied molecular orbital studies and molecular dynamic studies in progress. So based on these results, we may have a better structure with potent anti-biofilm activity. Then with our collaborators in our chemistry department, we are planning to synthesis for that particular structure. We are trying to move for ethical clearance. So this is our
Okay.
aim.
After the identification of this chemical, could we get the chemical from the commercial shop?
For commercial, we will go for process and product patent, and they will move on.
Okay.
That's what our.
Okay.
So this is what our research. We are taking the essence from the nature. That's the point.
Very nice presentation. Okay, scholars, any doubt?
Sir, I have a doubt. I'm Dr. Vinole from the Department of Zoology. Actually, our scholars, they are working on a protein called lectin, and they are just going for nano conjugation studies.
Okay.
So, is it possible to do docking studies with our lectin?
Your lectin. Yeah, it is possible, madam.
Okay.
Yeah, it is possible.
Okay. Sir.
But the structure should be available.
The only thing is you should have a structure of your both protein and compound ligand. That's all. If you have it, you can perform docking, whatever it is.
Yes.
Okay. Shall we wind up? Miss Stella?
A pleasant good morning to everybody. It's indeed my pleasure to thank you all in this pleasant day. First of all, I thank Lord Almighty for helping us to have such a wonderful program in this fine morning. Let me thank Dr. M. Karthikeyan, Assistant Professor and SCRB, Staff Fellow, Department of Zoology and Microbiology, Nagercoil College, MK University. He explained with medicinal plants and the extracts how he has performed his activity with all the plant parts and polymers. It's so beautiful, and the documents he has presented is with the graphical representation, and purely he has selected the data, collected the data, and it's highly informative. It's really triggering all the scholars to do definite research work and perform better in the coming days. Explain the uses of polymers which are natural and useful to us in many ways, especially in medical field. Explain with beautiful representation also. Really, it's worthy.
Thank you.
I thank you, sir, on behalf of the Department of Zoology, Holy Cross College. I thank the principal, HOD, professors of various departments and colleges. I thank the zoology department specially once again and the scholars. Thank you one and all.
Thank you. I thank Renuka madam for this knowledge sharing opportunity. Thanks a lot.
Thank you.
Thank
Thank
you.
you, Mr. Karthik. In your busy schedule, you have accepted this one. Thank you very much.
Thank you.